So the topic of revision for this video is mode of action of antibiotics, which cause damage to the cytoplasmic membrane. So polypeptide, the antibiotics that cause damage to the cytoplasmic membrane are the polypeptide antibiotics. They're produced by Bacillus species. They adversely affect the normal permeability characteristics of the cell membrane of the target organism. The examples of antibiotics included in this category are polymyxins, gramicidine, tyrosidines. So the polymyxins are effective against the gram-negative bacteria, and while the gramicidines and tyrosidines both are more effective against the gram-positive bacteria. These antibiotics, they are bactericidal because their bactericidal effect is due to the leakage that they cause from the cytoplasmic content of the cell that they are attacking on. These antibiotics, they have limited application in chemotherapy because they cause toxicity to the tissue. These two are the structure of two antibiotics, the tyrosidine A and the gramicidine S. We can see that all the amino acid are connected to each other. The amino acid configuration is of L type in both these cases, except for the ones that are specifically marked as D. So these phenylalanine wala ones are D configuration, while all other amino acids are in L configuration. Polyenes are the kind of antibiotic agents that have a large ring structures and they have many double bonds. The two kinds of polyenes are nystatin and amphotericin. The nystatin is obtained from Streptomyces neuresi and the amphotericin is obtained from Streptomyces nodosus. These polyenes, they act upon the cells that have sterols in their cytoplasmic membrane. They act on fungi, including yeast and other animal cells. The polyenes do not affect bacteria, so they affect the fungi. Mode of action of polyenes is that they increase the cell permeability. The next mode of action of antibiotics is through the inhibition of nucleic acid and protein synthesis. So they either inhibit the nucleic acid synthesis or they inhibit the protein synthesis. So the first antibiotic in this category is streptomycin, which is an aminoglycoside antibiotic. Streptomycin is obtained from Streptomyces griseus. This organism, this bacteria, was isolated by Scharf, Burgi, and Waxman, and they reported its antibiotic activity in the year 1944. Streptomycin inhibits organisms that are resistant to the sulfonamides and penicillin. So the antimicrobial or antibacterial spectrum of streptomycin includes gram-negative bacteria that includes Francisella and Francisella tularensis. Some organisms in the Salmonella group, several species of Mycobacterium, also including Mycobacterium tuberculosis, the causative agent of tuberculosis. Streptomycin's highly purified form are non-toxic to humans and animals when given in small doses. They have a cumulative detrimental effect on specific regions of nervous system when it's given as a medication for a long period of time. Aminoglycoside antibiotic, they have three components that are all linked together glycosidically. The three components of these aminoglycoside antibiotics are N-methyl L-glycosamine, streptose, and streptidine. The example of aminoglycoside antibiotic, apart from this streptomycin, the two other forms are kanamycin, that is obtained from Streptomyces kanamyceticus, and the other one is neomycin, obtained from Streptomyces stridei. The mode of action of these antibiotics are that they combine irreversibly with the 30S subunit of mRNA, and so due to their combination due to their combination with the 30S subunit of mRNA, the normal synthesis is disrupted of the protein. It is disrupted. Next antibiotic in this category are the tetracyclines. The tetracyclines, example, fluorotetracycline, oxytetracycline, normal tetracycline, doxycycline, and minocycline. They have the similar biological and chemical properties, all these examples of tetracyclines, and they are all grouped together because of their similar biological and chemical properties. These all are grouped together under tetracyclines. We obtain fluorotetracycline from Streptomyces aureofaciens, oxytetracycline from Streptomyces rhamnosus. The tetracyclines are a broad spectrum of antibiotics. The cross resistance of bacteria to tetracycline is common, and the tetracycline are bacteriostatic in nature. They hinder the growth of bacteria. The hydrochlorides and bases of tetracyclines are dry powder formed, and these are extremely stable in nature. The solution form of tetracycline is uh, less stable. Examples are chlorotetracycline and oxytetracycline. These tetracyclines, the solution form of tetracycline, they retain activity for 3 weeks, so for more than 3 weeks. Tetracycline have low order of toxicity. They are readily absorbed from intestinal tract, so which makes them effective when they are administered orally. So we can administer them orally as well because they are easily absorbed by intestinal tract. The mode of action of tetracycline include interference with the binding of amino acyl tRNA to the 30S ribosome. So due to this binding of tetracycline to the amino acyl tRNA of 30S ribosome, the protein synthesis of this cell is inhibited. Next antibiotic in this category is the chloramphenicol. Chloramphenicol is obtained from Streptomyces venezuela. They are a broad spectrum antibiotic and they act against both gram-positive and gram-negative bacteria. Chloramphenicols are bacteriostatic in nature. This is the structure of chloramphenicol, which contains a nitrobenzene ring. This is a nitrobenzene ring with the non-ionic chlorine. So these chloramphenicals have a very serious side effect of blood dyscrasias, which is a medical condition of hematologic disorders. That is basically disorders of the blood. So that's why they have limited hematologic disorders. The chloramphenicol act by combining with 50S ribosomal subunit. This combination of chloramphenicol with the 50S ribosomal subunit block the transpeptidation and translocation function associated with the site at which they act on. Next antibiotic is erythromycin. Erythromycin is the chemical class of antibiotics that is macrolides. It is obtained from Streptomyces erythros. These are active against gram-positive bacteria, gram-negative bacteria, and the pathogenic spirochetes. Erythromycin resemble penicillin, and they are active against organisms that have become resistant to the streptomycin and penicillin. So erythromycin is prescribed to patient with allergies when penicillin is indicated. So basically, the people who are allergic to penicillin, they are given erythromycin instead of penicillin. The structural aspect of erythromycin includes uh, it includes a large lactone ring that is linked with amino sugars through the glycosidic bonds with each other. Mode of action of erythromycin includes binding to the 50S subunit of ribosome, and due to this binding into 50S subunit of ribosome, transpeptide and transpeptidation and translocation of the protein synthesis are blocked. So this is it for this video. I hope it helps. Thank you so much.